All right. So what I said was 20, 30 questions for honors biology. Honors biology. It's going to be focused on the versus AP biology. It's going to be focused on simpler questions in the sense that they won't involve the depth of AP biology would involve any particular subject. But I expect that you'll be able to analyze an experiment. I expect that you'll be required to analyze a graph. And we talked about the kinds of graphs that you expect to see and tables, right? So you need to be able to get that straight. So again, the key here is that you're going to be applying your knowledge on some questions. Not all of them, but on some. Are we okay with that? Understand that? All right. All right, so tomorrow's your test. And this is for AP. Be different for honors, but so for AP Bio, what is we're looking at cell structure. And function. Unit 2. You should have gone over AP Classroom, should be done by now. So what have we done for prepare class notes? The book should have been read. Chapter reading's done. Oh, it's not showing up on the screen. Waiting for this thing to do its thing. Chapter reading should be complete. And notes done. Activities should have been completed. Study guides, etc. And there should have been AP Classroom done. And today we're just going to go over what it is, and any questions you might have concerning any of the topics. I'm going to go through each of the question types and what you can expect tomorrow. So tomorrow will be a multiple choice exam. With a limited curve. It should be, we spent so much time in it, I really don't think there should be any curve at all, but we'll see how things go. You can expect, I have 200 questions to choose from. It's going to be, I think we have an hour. Don't we have an hour for the test? So that's 60 minutes.
Yeah. We just have 54.
Or 54. So let's say 50 minutes. So at a minute a question, I should be able to give you 50 questions. But if I make it 45, I think that's going to be closer to what you're going to see in AP on the AP test. So let's just go with 45 questions, and that give us a buffer in case. But that means you have very little time to get in, settle down, grab your answers, and go. You'll have to fill up the whole period with trying to answer the questions. The questions will be short. There will be paragraphs involved to read, experiments to analyze. They're going to be based on the following, and I'm about to break it down right now. So you should know what it means to the difference between active and passive transport. Yeah,
What does the symbol mean under 45?
that means paragraph.
Oh,
So in active transport, you go from low to high. In passive transport, you go from high to low. Now, when you're thinking about active and passive transport, what you got to think about is that you have a situation where you have a lot of particles on one side, and if they have a barrier, they can't let them go through. What's happening with these particles, whether it's a gas or liquid or solid? But let's worry right now about liquids and gases. What do you know? What are these molecules going to be doing in this container?
like spread out.
These molecules or atoms, these particles, what will they be doing in the container? Say what? Moving. Yeah, they're moving. They're constantly moving. All things are moving all the time. They all have kinetic energy. So they're constantly moving. So if this side has a lot and you open a hole in it, you put a hole in this, or even just a couple holes, what's going to happen?
They gonna spread to the next.
They're going to move themselves just randomly, just vibrating, moving randomly, bouncing around. You're going to go from what your situation you have here to a situation. Oops. Give me a second. I think it's easier to do this than try to. A situation you have there to a situation more like. Okay. More like this, right? So particles are going to move to the other side, the opposite side. And they're going to spread out. These are kind of big dots, so let me make the dots the same so it doesn't look confusing. So, yeah. Yeah. So you go from empty on one side and full on the other. So in other words, just to be clear, you go from high on this side to low on the other. So now it's still high on this side, but less low would be a thing maybe. So, I don't know, it would be less low. All right, so not quite as low. Let me just, I don't know, that inequality seems wrong to me, so let's say less low. Less high. This is high. This is low. This is less high and less low. What happened? They moved across the membrane, the separator, and they became more equal as they're becoming less high and less low. Does that make sense? Anybody have a problem with that before I move on? All right. So finally, at the very last stage, what do you expect to see?
Reverse, like more gas on it.
Well, no.
Oh, yeah.
You expect to see, oops. You expect to see equilibrium, equal right and equal left. So we call that equilibrium. Have they stopped moving? No, they have not stopped moving. These things are always moving all the time. So, with the movement of the molecules going right at equilibrium is equal to the number of molecules going to the left. Here, most of the molecules are going to the right. Some are moving to the left, but most of them are moving to the right. So eventually you get an equal amount on both sides. When you do have equal amounts on both sides, everything else being kept constant, the particles are moving to the right and to the left at the same rate, leading to what we call dynamic equilibrium. They stay the same because they're moving in both directions at the same amounts. All right. Yeah.
Whether it be active or passive, they're both going to eventually end up in equilibrium.
No. Active transport, there's no guarantee you're going to be at equilibrium. In fact, often you're creating a concentration gradient. You're actually creating disequilibrium. That's the thing, is that this is distributed chaos. This is organized. This is more organized. This takes energy. Because to get everything in one place, we call it organization, correct? They take all the socks and put it in a pile. It takes energy, correct? To have them distributed randomly in any space, there's no extra energy required. That's nature. That's the natural course of things. That's how all things want to be. Remember, the universe wants to move towards chaos. It wants this. This is something life has to create. A concentration gradient. All the glucose on this side. All the sodium here. All the lipids here. All the proteins here. That takes energy. That's organization. That is organization, and that takes energy. Yeah.
I don't know, but in this number, like, why is it considered like chaos even though it's equal?
Well, chaos doesn't necessarily mean unequal. It means it's spread out. Like here, isn't this much more organized than this? This is just spread out. Right? They're spread out, but just like, think of organizing papers. Some of your teacher assistants, we put all the papers in little stacks everywhere, right? What happens over time if we don't do that? It spreads out, doesn't it? We call it messy, means they spread out. This whole room, right? You've seen it. There'll be papers everywhere. Already we can see some papers starting to spread. That is disorganization. To organize it, what do? Collect them and put them all in one place. Well, this is organization, right? This is chaos. All right, so dynamic equilibrium is a reaching of this equal state. That's just going to naturally happen. That requires no extra energy. That's why we call it passive transport. Just what's the kind of passive transport that we're talking about here? What is this called? What is this called? What is the word for this? I heard the word diffusion used. You're right, it's diffusion. It's a word called simple diffusion. Yeah, it's just the idea that you have a high concentration. If it can move, it'll move and spread out. And you'll get equilibrium, a dynamic equilibrium. We call that simple diffusion. And the examples I've used since your 9th grade year is that you have a person here and they let out a fart, and then that fart starts to spread. That gases, those particles of stinky gas spread out, and eventually someone far away as the other side of the room, all over here, will smell it. But if you're the unlucky individual that's sitting next to the person who farted, you smell it a lot stronger. If I farted, someone back there might smell. There's a hint of something in the air. I'm not sure, it's unpleasant, but where someone over here would be, gee, I'm getting, it would be really awful. I'm trying to fart. It's not working, as an example. It would have been a great. No, you wouldn't make it to the door. And to get to the door, you have to pass by me, which puts you right through the middle of the what? It would put you in the middle of the what? You have to breathe. That doesn't help. You have to hold your breath completely. You have to walk through this. What is this? This is diffusion gradient, but what's this? If you have to walk through this to get out the door, what is this? It's the center, but it's the place where what? It's the strongest. It's the high. So this is where the concentration is the highest, high, and this is where it's the lowest. And this, from high to low, is called the concentration gradient. The concentration gradient. It's going from high to low. That's it. We want to take advantage of that. That's how life does. By creating compartments, we can create a high concentration, and then we can control, the cell can control the openings. Then we can control the flow. That's what we do in our bodies. Every cell on the planet does that. It creates little compartments. Bacteria create one compartment. We create many. And we're able to control the concentration of different particles in those compartments, and then control when we let them go and how fast we let them go. You do that with your cell phone all the time. You have this little thing, this little flat thing inside your cell phone that you've probably never seen. It's a thing called a battery. And inside a compartment in that battery is an ions, a bunch of negative ions. And inside another compartment in that battery is a bunch of positive ions. And then you shoot what you do is you connect the circuit by pushing a button, the on button, and the phone turns on. The electricity flows. It goes from high electrons to low electrons. Flows. Push another button and turn off, the flow stops. To recharge it, you have to put energy into it. To make it go the opposite direction, to recharge your battery, what do? Plug it in. What you're doing is what? Making the electrons go against the concentration gradient, going from low to high. Creating organization, organizing that battery. Taking all the electrons, putting them in one place, disturbing the dynamic equilibrium, charging it up, then you get to 100%, most of the electrons are here, and let it go again. And then what happens? It flows slowly, and then you turn on, it flows quickly, you turn up, you use even more. You call that measurement current. Yeah. That's a great question. So the electricity comes from the power plant, where they took, and even though this doesn't have to do with a cell, it will have to do with ecology later. So the electricity in this example of batteries, you can take and make three different kinds of, we use two in Ohio, but we have the nuclear power, which is using radiation to boil water. That's all you're doing. You're boiling water. That's the whole point of nuclear power. Or you could use coal, which we also use coal in looks like more like this. And coal burning power plants also boil water. In both cases, you're boiling water. You're increasing the kinetic energy of water to a gas, and that gets put into a pipe. That steam, I think this pencil's too thick. That steam pushes this thing called a turbine. A turbine is like a water wheel. As the steam hits the fan, what happens to the fan? It starts moving, it starts turning. And these turbines are big. They're as big as this room. They're bigger than this room.
The big white thing?
What the?
That in the middle of nowhere?
Oh, you're talking about windmills? Windmills are the same thing. The whole point is to turn the wheel. That's what we need to do. We can put hamsters on a wheel and turn the wheel. We could do that. You need a lot of hamsters. Or you can use the wind to turn a big wheel, the big windmill, correct? Right? That's all we're doing there. Or you can boil water using coal and push the steam to turn the wheel. Or you can boil water using nuclear power and that turns the wheel. You can use a waterfall, like in Niagara Falls. You can use the water falling to turn the wheel. I don't care how you turn the wheel, but all of it's about just turning the wheel. If you can get the wheel to turn, that wheel is connected to magnets. Just a bunch of magnets. Thank you. And now as you turn magnets, and inside the center of the magnet is a copper coil. So these coils, I don't think I have a copper color, so you just have to go with me and say that's copper. You have these copper coils. Copper conducts electricity. They're good conductors. So this is copper. So you spin the magnets around the copper. Just spin them. That's all you're doing. How do you spin them? You use the wheel. What do you use to push the wheel? Hamsters. Not a good idea. Wind works well. You can use water, like in waterfall. You can use burning coal and boiling the water, creating steam. You can use it. Nuclear power can do it. Some people even use wave power, the power of the waves in the ocean or lake, to move the wheel. But no matter what, you have to move the wheel. And the wheel, this is called induction. If you move the wheel, then that pushes electrons. The magnets push electrons. You alternate positive and negative. Magnets have positive and negative. They push the electrons down the wire. Really big, thick wires. You see them on the big power ones. You can see it right there. There's a big one going right to that transformer plant. And that goes to your house. Eventually, there's all kinds of intermediate stages you have to down. This is like 10,000 volts, something like that. And you have to take that down to something like 120 volts, which is what you have coming out of your house. So these wires come off and go into your house. And in that house, it's really not there we go. In that house, you have a plug. Bless you. And you plug the wire. You plug your wire into that plug. The electricity goes into your phone and pushes the electrons. So these big nuclear power plants and burning tons of coal and water and the wind blowing all those big turbines, all that so you can charge your phone, turn on the lights. Right? Everything's running through that electricity, running through those wires. So that's a really great question. All of it is about high to low, isn't it? High to low, high to low, high to low. So concentration gradients, they run just about everything. And the wind itself is created by high pressure to low pressure. All right. So that's simple diffusion. That's concentration gradient. Now, what about things that don't pass the membrane easily? What about, I'm going to create a membrane, just a line, if you don't mind. I don't want to draw those heads. If I have a lot on this side and not a lot on that side, what passes through what molecules would pass the membrane easily? What passes easily? Small nonpolar, right? And small polar is exactly right. The easiest is small nonpolar, but small polar also pass. So they pass easily. That would just, what would the examples of that be? The example of small nonpolar that everyone should know for sure in this class is CO2 gas, oxygen gas, and nitrogen gas. Nitrogen is only really important when you're diving or some other situations, but as far as your body's concerned, these gases are constantly flowing in and out of living systems. What's this example of small polar? Small polar, what would be an example of the small polar that you pretty much absolutely, that's right, H2O. You absolutely need to know that, small polar. So small polar, you have H2O, small nonpolar. So assuming it's these particles are any of these, it could be any or all of these, what direction would things flow? Left to right, right to left. In the case that you see right in front of you. Left to right. Why left to right? High to low. This is high, this is low. You can see them. So you will see questions where you just see dots. You see dots on one side, or they will say high. They'll actually tell you this is high and this is low. All right. We call this passive. This is simple diffusion. All right. Now, what about things that won't pass? What if you have a membrane? You have things on this side. What kinds of things don't pass? All right, so large molecules, true. Yes, and what else? It's something you should know. Absolutely does not pass. Absolute, no question. Say what? No, fats have a hard time even being dissolved. We have to create phospholipid things called vesicles to transport them. So in a sense, that's a complicated answer. But they do pass through the membrane easily. Fat oils and lipids will pass through, even if they're big, pass through pretty easily. The hard part's getting them to the cell. So the question is, what does not pass that's in water? It's already dissolved in water. Because remember, the solvent is water. Large molecules in general are not going to pass. So yes, I agree. It's large polar would be one like sugar. Right, like glucose. Sucrose is a big one that won't pass. It's a disaccharide. It's just one doesn't do well. But what is? Give me something else that you're missing. No, hydrophobic's gonna pass through the membrane. Remember, it's hydrophobic. Hard parts getting the hydrophobic substances to the membrane, but once it's there, it'll get through. No, well, that's true too. Large amounts of water is not easy to get through quickly.
Maybe like something that we exactly.
You're missing that. Okay, you're all going down really complicated answers. I'm looking for a simple answer. You're missing it, so I'm gonna give you a minute to answer it. Ions. Yeah, that's it. Whoever said ions is right. Yeah, ions is the thing that will not pass. They're small. They're tiny. They're tiny particles. They won't pass easily through a membrane. So absolutely, ions are something you got to expect. Ions. So large molecules in general, polar or nonpolar, large-ish nonpolar molecules can get through, but even really large nonpolar proteins, mostly nonpolar proteins, are still going to have a hard time getting through that membrane because they're big. It's going to disrupt the membrane. So that goes to phagocytosis. That goes to pinocytosis, getting large amounts of water into the cell, large molecules in the cell, large quantities of things in the cell. So we'll look at that in a minute. But do not pass large molecules and ions. So let's say these things are ions or large molecules. So they would be proteins. It could be something like sucrose. It could be something like ions. Any of these things is what we're talking about in this model that's in front of you. What's going to happen? Is it going to pass? Are they going to pass? What's going to happen because they can't pass? You would need a transporter if you wanted to get them across, but let's assume for a minute that there is no transporter here. Well, no, the question is not how do we get it across. If I set this situation up, you guys are answering correctly. I want to point that out. But that's not the question I'm asking. I'm asking if we have this model right now, what's in front of you? This side, these cannot pass through the membrane. This is, I don't know, let's say 10%. And this is 0%. Let's get rid of this and just call it 0%. So there's 10%, and this is 0%. Ions, proteins, sucrose, whatever. If this is 0% that, what is it? What's the rest?
Water.
It's water, so it's 100% water. And what is the other one? 10%, that stuff. What's the other? What percent is it over here? What's 90% what? Water. It's in solution. It's in water. Water's the solvent of life. So, okay. So 10% sucrose, ions, or proteins on one side, 0% on the other. It's not going to pass. What's going to happen? Osmosis, correct. So because the ions can't pass, osmosis is going to happen. And you have two things you have to know, two ways of looking at it. Your level of education, you should know both. You should understand both ways of looking at this. If the substance is not passable, then water has to move. And in this case, which way is water going to move? Just before I even go on to explain, which way is water moving? Right to left. How do you know that? It's high to low. Water's high to water's low. Why? Why would it do that?
Because they got so they
Oh, no. Why would the TV turn off? Like, why is that happening? I didn't push any button. It's a setting. I know it's a setting, but which setting? Where is it? Do I have 2 hours to figure this out? Huh? I know, but I don't know where the settings are. I've looked at the settings. I have every setting I know I had the tech guy in here for half an hour to figure out how to get the sound to work. He finally got it to work. After 5 menu layers, he found the one setting that had the sound off. All right. So, did he go off again? It's just having a hard time. Life is hard for it. So, anyways, osmosis that's right. Osmosis, when the ions, the sucrose, the proteins, when there is no transporter protein, then they cannot pass, so water will move. We call that osmosis. Water will go from water's high to water's low. So it is a diffusion, it is a form of diffusion. Osmosis is a form of diffusion. It's the diffusion of water. The diffusion of water is called osmosis. So water goes from its high to its low. Is that clear? Water moves when the solutes can't. So that brings us to a discussion of hypertonic, hypotonic, and isotonic. So when you look at substances, there's 3 states that you really have to concern yourself with. When we use the word tonic, What you're talking about is the stuff that's dissolved in it. What's dissolved in it? And the hard part here is that it's all relative. Everything we're talking about is relative. So it can be very confusing if you're absolutely not sure of the context of the question. You have to read the question, think, reference, find a reference. Where am I? Where is it? Where are they saying it's higher? Where are they saying it's lower? Most of the time it's obvious. Sometimes it can be very tricky. So, if you have in scenario 1, let's look at a hypertonic solution. If they say I put a cell into a hypertonic solution, I put a cell into a solution, and it is 90% sucrose, and inside it's 10% sucrose. Where is this solute higher? Outside. So the solution is hypertonic. The solution is hypertonic. All right, the solution is hypertonic. Now, again, why do I say the solution is hypertonic? Because I'm talking about the outside. But if somehow they phrase it really oddly, if they decide to try to trick you, which I don't know if they're trying to trick you, if they're just stating it oddly, I don't know what. But if they talk about the inside of the cell is hypertonic, then the 90 would be inside. Does that make sense? So hypertonic just means it's higher salt concentration. Higher than what? Than the other side. Whatever that other side is. Does that make sense? Yeah. Generally, they're talking about the solution you're putting in it. So usually that's the case. But I've seen crazy outside. But I've seen crazy questions, so you have to be careful. I'm telling you, just read. Read and think. Yeah. If the solution is higher on the side you're talking about, in this case it's going to be the outside. If it's higher, then it's hypertonic. So what's the inside solution? Hypo. The opposite's happening on the other side. So one or the other. See how relative it is? It's very relative.
But if they just ask which one is the highest, we just know it's hyper.
Yes. Higher means hyper. Hypo means low. When you're hypoglycemic, you have low blood sugar. You're hyperglycemic, you have high blood sugar. Hyper means high. Hypertension means you have high blood pressure, etc. Hyper means high. So here, in what would if this doesn't pass, which we're going to assume it doesn't because it's sucrose, some things they're going to expect that you know. Most questions, they're going to contextualize it. They're going to tell you it doesn't flow, or they're going to draw an arrow and show it not passing. In some way, they're going to give you clues in the context on whether the stuff passes through the membrane or not. You could have sucrose pass through the membrane if you had a transporter that would allow it to get through. But in this case, it's not going to. So what's happening in this solution, in a hypertonic solution? Water's going to move. Water's going in, he says. Water's going out, he says. Do you agree? Is water... Think.
It'll go out and it'll shrink. Yeah.
It'll go out and it'll shrink is the right answer. Yes. But the question is why? Why is water going out? So there's two ways of thinking about it. Two ways. You should know both because they could...
because it's 90% water.
That's right. That's right. So if it's 10% sucrose, then it's 90% water inside. And if it's 90% sucrose outside, then it's only 10% water outside. So H2O is going to go out because it's going to go from its high to its low. That's one really great way of thinking about it. But they might ask you a question, is put in a hypertonic solution. They won't tell you. They say, what is the direction of water? What is going to happen? Well, water's going to flow out. The easier way of remembering it is that if it's hypertonic, water's leaving. It's shrinking. If you put something in a high salt or high sugar concentration, the sugar or salt can't pass through, then you're gonna get shrinkage. So in a hypertonic solution, this cell, in this case we're talking about a cell, this cell shrinks, shrivels up, whatever word you want to use. Until, because the water's leaving, until they've reached some kind of dynamic equilibrium. All right. Now, what about an isotonic? What does iso mean?
Like, the same amount of protein.
That's right. It's the same. Iso means the same. That's what iso. Iso means the same. I think it's Greek, actually, but I'm not sure. You have to talk to Reverend Greg on that one. So isotonic, it's the same. So you would expect if somebody's telling you it's an isotonic solution that you're going to get, 50. No, let's not use 50 because people are going to think it's 10% and it's 10%. So what's going to happen if the solute can't pass through the membrane? What's going to happen? Nothing. Water will move. Water will still be moving. It'll just be moving in both directions. Okay? And here water's moving in both directions, but water's moving a lot less in than it is moving out. Does that make sense? The majority of the flow is out. So it ends up net movement is out and it shrinks. That's an important idea, but there it is. So there's nothing happens. The cell stays the same size, no change. So remember, dynamic equilibrium means they're moving in both directions, in and out of the cell. All right, it means they're moving in and out. But when you have a change in shape, it's because the net movement, there's more movement in one direction than the other. If the net movement's out of the cell, then the cell shrinks. Right? Make sense? All right. The last example here is hypotonic. What do you expect to see here? A swelling of the cells, which you'd expect to see, and that's what happens. So here you'd have 10% outside and 90% inside. And so we'd have 10% water inside and 90% water outside. So what you'd expect to see is the water's going to go in. And in fact, it'll go in, it'll keep going in. Mostly, some will go out, but mostly it'll come in until the cell blows up, until it bursts. So here it actually will swell. And sometimes, not all the time, but sometimes it swells so much it bursts open and out comes the contents of the cell. Lysis, yes. This is lysis. This shrinking is plasmolysis. Lysis means to break apart, to break up. Hydrolysis is to break up using water. Plasmolysis is when the cell shrinks so bad that it breaks apart. It just stops functioning.
Isn't that only in animal cells because the cell wall holds up?
The cell wall helps a lot, but eventually, if you a plant cell will droop until finally it also will collapse. But it does have cell walls to help it not completely plasmalize. Lysis certainly is very difficult in plants. Plants will have that cell wall and that will hold it. It won't burst. So lysis doesn't happen very easily at all in plant cells. Okay, or for that matter, every eukaryote except for animals, lysis is difficult to happen because those animals, almost every eukaryote and prokaryote has a cell wall. Cell walls help these things not lyse because of water pressure. But animals don't need that because animals control their water pressure. They don't need a cell wall. We run around, we get too hydrated, we stop drinking water. We get wet, we go somewhere where it's dry. A tree can't do that. So there you go. That's hypertonic, isotonic, hypotonic. That's most of the passive transport concepts, simple diffusion. Now, what about stuff that is going to try to get through the wall, the membrane, that we need to get through? What about this stuff, like somebody mentioned earlier, a large polar molecule? What is this large polar molecule I just drew up here really quickly, sketched?
Carbohydrate.
And carbohydrate is correct. It's glucose, usually. Could be fructose, but usually they draw glucose this way. So you see a symbol like that, usually you're thinking automatically, don't assume, read, contextualize, but normally you're thinking glucose, right? If you see a symbol like this, you might be thinking ribose, right? Just thinking, you'll have to see by looking at it, but those are the kind of two shapes you should be solidified in your mind. So out here is glucose, or let's look at glucose. Let's forget about, because that leads us to a discussion about diabetes. So let's go ahead and use that. Let's talk about glucose. So if glucose needs to get through the membrane, how do we get it through the membrane? It needs help. What do we call it when we help things across the membrane? Right, it's using a transporter. What is that called? It is, but all of it's membrane transport. Diffusion is also a form of membrane transport. It's just random. This is something where membrane transport needs help. You're right, you'd use a transporter protein. No, it could still be passive. What's another word for help? Assist is correct. Another word for help? Facilitated, yeah. Do I have it on the board? No. Facilitated diffusion. Facilitated diffusion is passive. In other words, it requires no energy to help the things across. No extra energy. So all I got to do is put usually a transporter, as somebody mentioned earlier, some kind of protein, a tunnel, if you will. I'm just going to simply draw something like this to allow the particles to transport in. That's it. So just that simple a movement is considered passive transport. That's a facilitated that's a protein channel. Also known as a transporter.
You got 3 minutes.
All right. I'll try to use all 3 minutes to get you as much as I can, as much help as I can. So this is a transporter protein. It's a facilitated diffusion. But what about when we use transporters to cross the membrane for things that we don't? What about when we have some kind of transporter? It can't be just a channel. It can't be just an opening. It has to be something more complicated. Some kind of sodium, potassium, ATPase pump, for instance, where we're going to get this ion to move in this direction. What kind of movement is that? Active. So this is active transport, because now we're going from low to high. So this is active transport. And we're going to talk later, after the test, we'll talk about some really specific types of transporters. But right now you need to know generally. I'm going to make sure I don't ask you anything on a test where you have to memorize a specific type of transporter. The diagrams will tell you much. You have to understand what active transporter is. This requires what energy? This requires, I see your hands, give me a second. Energy. It requires energy in the form of ATP. Requires energy, yeah. Yeah, low to high, yeah.
So if it's low to high, then it's active.
Yeah, because you have to push it. I keep using this example, but just go back to the simplest example. A rock at the bottom of a hill. Does it take energy to put it to the top of the hill? Yeah, because you're going up. But if it just rolls down, you don't require any extra energy.
Right, okay.
All right, so just think of it that way. Try to keep those simple examples in your mind on these things. Know what each organelle does. Know that. Know the how we get proteins out. I will not ask you transcription translation questions, so don't panic. Like Golgi body, what does it do? What does an endoplasmic reticulum do? What does a rough endoplasmic reticulum do? What does a ribosome do? What does a ribosome in the cytoplasm do differently from a ribosome in the rough endoplasmic reticulum? What does a nucleus do? What does a nucleolus do? What does the cell membrane do? What does a mitochondria do? What does a chloroplast do? What does a lysosome do? What does a vacuole do? All right? Know those organelles. All right? And you should be fine on this test. I'll tell you right now, you're looking at specific protein, transporter proteins, again, concentration gradients, again, I'm looking at the questions right now. There's some questions that are reading some charts, RNA, ribosomes, all right, signaling. I'm going to try to minimize signalings. Aquaporins, which is, again, just a way to get water into the cell a lot faster. Bacterial structure. What does it look like? What does a bacteria look like? A really crazy question I'm not putting in the test about signaling. That's for next test. There's a chart reading a table, types of cells, fungal cells versus eukaryotic or animal cells, that kind of thing. Mitochondria, prokaryotic cells. What's the same in prokaryotic cells versus eukaryotic cells? What's different in that? This is all in the chapter reading, so it's there, and in the AP classroom. What ATP, how do you, when active transport, what would you expect to happen? Another diagram about transporting cells. Another diagram about plant cells and all their structures. What is all more about the plants and their structures. Dialysis tubes. Where would you expect to see water mass gained or lost in different solutions? More dialysis. Again, it's you have to read, but it's pretty straightforward. Simple diffusion. Looking at pictures of plant cells that are in different solutions. I'm not asking you this question. I don't even know why they put it in there. I asked them to give me specific questions. Prokaryote, eukaryotes. So there you have it. That's the kind of stuff you're going to have on the test. I'll have to pick the questions I'm going to put on your specific test and get rid of the other ones, and you're taking the tests tomorrow.
Is it ready today?
Is it ready? Yes. I will. I have to edit it. If I had to give it to you today, right now it'd be a 26-page test.
No, I'm not saying right now, I'm saying about 7th period.
7th period, yes. It will be ready by 6th period because I have a student coming in to take his 6th period. I know. Either is Rachel. She's coming in 6th to do it.
I just have a question on this.
Which one's it? Which direction would an arrow need to point to show the movement of molecules during diffusion? So what would you think? To the left, that's correct.
But,
High to left.
okay.
High to left. I don't like it because they didn't put the two boxes next to each other, right?
Okay.
But it's always high. If it's what are the key words here? Let's look at it. We're going to look at diffusion, which means passive,
Mm-hmm.
right? Diffusion is always from high to low. Where is it high? Well, it says high. Is it low? It says low. High to low. That's from right to left. Because it says which direction would the arrow need to point? It needs to point to the left because it's going to go that way.
Oh, okay. That makes sense.
I was gonna ask, is there any situation or like opportunity for like, I guess for lack of a better word, cells that are not like, or like ions or whatever is like not good for you? Can they like camouflage themselves so that they can get in?
Ions are just atoms with charges on them. So they're not, there's no camouflage, they just exist.
Okay.
We ingest them. We choose to ingest them, like when we choose to eat salty potato chips or
Mhm.
put salt on our food. So ions are in our blood. That's how they get in our blood. Okay. Now, once they're in your blood, what do? They're going to increase. They don't pass through the membranes easily, so water is going to quickly go into your blood because it's hypertonic.
Okay.
That's going to raise your blood pressure.
Oh.
So salt can be bad for you, but not in a way of that you're discussing.